Recall that enzymes are an example of proteins in the body. Enzymes are substances produced by living organisms that act as a catalyst. Catalyst means to speed up. In this case, enzymes are speeding up biochemical reactions to produce an end product. In this presentation, you'll be learning about how enzymes work and what happens when they stop working. Make sure you are following along with your own notes. Every reaction needs energy to get started. This type of energy is called activation energy. In this chart, the amount of energy needed is on the y-axis, and the time it takes for the reaction to occur is on the x-axis. The arrow shows the amount of activation energy needed to be able to drive the chemical reaction forward, changing the reactants into products. Without an enzyme present, it'll take longer for the products to form. Your first keyword is most. Catalysts help chemical reactions happen faster. This is done by decreasing the activation energy required for the reaction to get started. An enzyme is a type of catalyst. As you can see in this chart, the top lighter line is showing a reaction without an enzyme, and it's taking longer and needs more activation energy to get started. Whereas the bottom darker line is showing a reaction without an enzyme, resulting in a shorter time because it decreased the amount of activation energy that's needed. Your second keyword is reactions. An enzyme is a type of protein. These proteins act as catalysts to speed up chemical reactions. Your third keyword is in, I-N. Here is an example of the process of how an enzyme works. Up here, you see this green shape, which is an enzyme. The yellow shape over here is called a substrate. The substrate will be able to bind to the enzyme because it fits with the active site of the enzyme. This part right here is called the active site, and this is where the enzyme and substrate bind together. When they are binded together, this is called the enzyme-substrate complex, and this occurs during the reaction. When the reaction is complete, the enzyme releases the newly made products that the substrate had turned into. The enzyme comes out of the reaction unchanged and ready to be used again. Only certain enzymes bind with certain substrates. Think of this like a lock and a key. Only a certain key matches a certain lock to open something. The same goes for the enzyme and substrate. The substrate will only fit a certain enzyme's active site. Your fourth keyword is your. At the end of the reaction, the enzyme is unchanged and can be used again. As you see in this picture, the enzyme looks the same when the reaction starts and the same at the end of the reaction after the products leave the active site. Your fifth keyword is body. At the end of the reaction, the enzyme is unchanged and can be used again. As you see in this picture, the enzyme looks the same when the reaction starts and the same at the end of the reaction after the products leave the active site. Your fifth keyword is body. There are ways that will cause an enzyme not to work. The two main factors that cause this are change in pH and change in temperature. If the pH is too acidic or the temperature is too hot, the enzyme will denature, meaning to change shape. This will cause the enzyme's active site to change shape, so it won't be able to bind with the substrate anymore. Therefore, the reaction will not occur at the time that is needed. These pictures show how an enzyme can be affected by the change in temperature or pH. The active site will change shape, meaning it won't be able to bind with the enzyme. For example, this is why our bodies need to maintain homeostasis, to maintain body constant temperature and pH. If our body temperature gets too hot, meaning we have a fever, certain enzymes will not be able to catalyze reactions. This will eventually cause our bodies to shut down and we would die. This is why it is very important to get rid of a fever, even though it is our body's way of responding. Our sixth keyword is require. You will see that enzyme names usually end in ACE or ASE. The beginning of their name will tell what it does. Some examples are protease, DNA polymerase, and ATP synthase. Protease breaks down enzymes, and DNA polymerase joins monomers to make DNA. We will be talking more about DNA polymerase in the upcoming unit of DNA. ATP synthase synthesizes or makes ATP. There are two major processes you need to know. Both are catalyzed by enzymes. The first is dehydration synthesis. In this process, two molecules are bonded together by removing a water molecule. If you look in the picture, you see a glycerol molecule on the left and a fatty acid on the right. The OH of glycerol joins the H of the fatty acid to make H2O. Your seventh key word is enzymes. The second reaction is hydrolysis. In this process, two molecules are broken apart or separated by adding a water molecule. This image shows an H2O molecule being added between molecules 3 and 4. The separated molecules then receive one part of the H2O molecule, either H or OH. Use this coloring guide to help you color the diagram in your notes. The protein which controls reactions in living things is the enzyme. The reactant of an enzyme called catalyzed reaction is known as the substrate. The location where the enzyme and the substrate bind together is called the active site. The enzyme-substrate complex is what is formed when the enzyme and the substrate bind together. At the end of the reaction, the substrate is changed into the product. So we've now concluded our notes about enzymes and reactions. You can make sure you have all of your fill in the blanks, and then you can use your notes to answer and complete assignments throughout this level.